All right, thanks for coming today and to this session. I'm excited to share with you some of the research coming out of my master's degree at the University of Hawaii, focusing on the Rumped storm petrel. And so, as all of you in this room, I'm sure know, seabirds are top marine predators that reveal the health of our oceans, and they've also been a keystone species to the Hawaiian Islands, facilitating the growth of our native forests by bringing essential nutrients like nitrogen to our soil. And because they have large ranges, forage on the sea and breed on the land, they encounter many threats. So I'm sure you've seen lots of talks on predators, and this is a huge threat here in Hawaii. Cats, mice, mongoose all predate on chicks, eggs, and adults. And humans aren't doing too good either. We have contributed to habitat loss. Light pollution is also a big threat to storm petrels, as well as marine debris, which can lead to entanglement and ingestion. And so the bird I'm focusing on today is the Rumped storm petrel. So before human contact, it was found on all of the main Hawaiian islands, and based on midden site record and historical and fossil record. But now colonies are limited, and there's only a few on a few islands that we believe that they're currently nesting on. And so in 2016, the Rumped storm petrel in Hawaii became endangered, and which prompted the funding for this study because there was concerns of inbreeding because of their low population size. And so due to their low population size, I predicted that there would be high genetic structure between the islands and relatively high rates of inbreeding. And so over the last few years, we've collected samples across the Hawaiian Islands and also collected samples from the Bishop Museum that are already available. And so, as you can see, samples came from carcasses found at colonies, as well as live birds that were captured near colonies, and also came from downed fledglings and bycatch individuals, so birds that washed up on the shores. And just to point out here on Oahu, we currently don't think that they're breeding there, but there was a couple individuals that washed up in the last few years, not during the breeding season. So they probably belong to another colony on a different Hawaiian island. And so we also sequenced these individuals to find out their population assignment. And so once I had the samples in hand, I extracted the DNA. I amplified that genomic DNA because a lot of the samples, like from the museum samples, had a low genetic weight. And then I quantified each sample, measured equal amounts of the DNA, and then used restriction enzymes to cut the DNA and size-selected fragments to be run on an Illumina HiSeq, so a next-gen sequencer. And then I received those sequences and performed bioinformatics, which is genetic analysis. For those who are not geneticists in the room, how I analyzed this genetic data was using single nucleotide polymorphisms, or SNPs. This is an example of a SNP, as you see in the circled in red. So these are lines of genetic code coming from different individuals. So here you can see this SNP. So some of the individuals have a base pair of a C or a T, and these differences is how we were able to measure genetic diversity and difference in similarities between individuals. And so because I think I received over 600 million paired end reads, I use different computer programs that are built for genetic analysis. And if you guys have specific questions of that pipeline, please come talk to me after. But just to give you a general idea of how I analyzed this DNA was that I did an assembly of the reads. I filtered for those SNPs. I created a mitochondrial genome and matched SNPs to the mitochondrial genome so I could find which SNPs were nuclear, which SNPs were mitochondrial. And I also did a population assignment, ran a principal component analysis, found FSTs between the island populations, as well as found inbreeding coefficients and ran a Bayesian skyline plot. And so one of the important steps with the SNP filtering, it ensures that we're looking at real SNPs and not just sequencing errors. So I used min and max coverage and an optimal minimum quality value so we could ensure that these SNPs are indeed real. And what we got was over 13,000 nuclear SNPs and 67 mitochondrial SNPs, which is a typical ratio because the mitochondrial genome is a lot smaller than the nuclear genome. And this is the structure plot that I created. And so here you can see that the individuals from the main Hawaiian Islands cluster in two different genetic groups, and most of the individuals from Kauai and the Big Island belong to that blue genetic group, and then the individuals from Maui and Oahu are mostly in that green genetic group. So there is some separation by island of where the individuals were sourced. And the same story goes for the principal component analysis. So you see the clustering of Big Island and Kauai individuals on the left-hand side, and then on the right-hand side, more the Maui and Oahu individuals. And I also found FSTs between the islands of which the individuals were sourced. And so same story, we see that there's higher differentiation between Kauai and the Big Island to the Oahu and Maui individuals, and similarities between Kauai and Big Island and Oahu and Maui. FST is a fixation index, so it ranges from 0 to 1, for those aren't familiar. 0 meaning there's complete mixing of individuals, and 1 meaning they're completely different, to give you guys a reference. And also, for inbreeding coefficients from the most sampled islands, the Big Island and Kauai, we found it ranged from negative 0.057 to 0.097. So this kind of averages around 0, which shows no evidence for severe inbreeding. And then also looking at the Bayesian skyline plot, so this is the effective population size over time with going towards present day. So 0 on the left-hand side is present, going 600 years back. And so from this analysis, based on mitochondrial data, we see that there's a steady decline in the last 600 years with a small expansion in the last 25. And the shading is a 95% confidence interval. So the effective breeding size, effective population size today, based on this analysis, ranges from like 100 to 1,500. So it's definitely a wide range, but gives us a better idea of maybe how many banded storm petrels are nesting in the Hawaiian Islands, but definitely more on the ground field work and needs to be done to confirm this number. And so also looking back at the structure plot and also the FSTs, so we see this, the Maui and Oahu individuals actually have a negative FST, so this says that they're more similar than they are different. So these individuals are probably coming from the same genetic group, same colony, but which is separate from those of Kauai and the Big Island. Because we don't think that there's one in Oahu, these birds might be coming from a colony in Maui, or another one that we just haven't sampled yet. And so some of you may be asking, what is the role of genetics in management? As there's no surprise that genetics is relatively expensive, so why would we do it? And this would be to answer questions of connectivity, are species migrating and moving within their range? We can answer questions about sex and species identification, and also address concerns of inbreeding. And so that is what we did in this study, because there was an inbreeding concern, because there was few known colonies for this bird, they're endangered and in low population values, or numbers. And so the good news coming out of this study is that there's no evidence of inbreeding, and that there may be 2 different genetic groups in the Hawaiian Islands. And so even though the banded storm petrels here don't look like they're vulnerable to an extinction vortex based on genetics, there's other threats that remain. So predator control and related management efforts are still necessary to continue to recover these populations because they come from the procelariforms, They are highly phylopatric. They return to their natal colony in which they were born. So they are a good candidate for translocation. And just overall, moving forward, it's just important to protect the rest of the bandworm storm petrels that we can find here in Hawaii, and less important to know where they're coming from. And so, in the hopes of protecting seabirds, if you find a downed seabird here in Hawaii, contact the Hawaii Wildlife Center, have their helpline and email. And I would not have been able to do this study without lots of support and partners across the Hawaiian Islands, many of you here today, so thank you for all the help.